In this lesson, we're going to talk about one of the key figures in the history of biology, Gregor Mendel, and how through his experiments he managed to develop a particulate model for inheritance. Here are your objectives. Now, before Gregor Mendel, of course, it was already observed that offspring resembled their parents, but one of the leading theories of inheritance was that it involved a kind of blending, where what was being passed down from generation to generation was being blended, much like paint or solutions would be. But Mendel, through his experiments, showed that blending was not the correct model, and instead something discrete was being passed from one generation to the next. There were discrete particles or units of inheritance that were being passed from parent to offspring. So Mendel was a monk living in the 1800s, and he did his work in the mid-1800s, actually while he was living in an abbey in what's now the Czech Republic. And he did his experiments on peas. That was his experimental organism. And peas were really good for his experiments because peas can self-fertilize, so you can cross or breed a pea plant with itself so that it's both the mother and the father. They also have a number of very distinct traits, and Mendel experimented on 7 traits like flower color or the color of the peas, etc. And they also allow you to very carefully control the parentage of peas. You can always be sure that you know who the mother and the father plant are by controlling pollination. So peas were a good choice. It's also important to keep in mind that Mendel was working before DNA or chromosomes were even discovered, and of course, therefore, before they were associated with heredity. So he was working in the absence of that knowledge. So what did he do? Well, he would look at each of these 7 traits one at a time. So let's look at the color of the peas themselves. Mendel would take 2 different colors of peas, let's say yellow here and green, and he would obtain these as true breeding peas. So what that would mean would be he would take a plant that only made green peas and he would self-fertilize it. He would breed it with itself for many generations so that all of the offspring were only green. That's what true breeding means. And then he would do the same with a plant with yellow peas, so that it was bred with itself, or multiple plants were bred with themselves for many generations so that they were only producing yellow peas. So he would take a true breeding yellow pea plant and a true breeding green pea plant then, and he would cross them. He would breed them together. And when he did that, he saw, as a result of that cross, only yellow peas were produced in the offspring. So already we see a problem with the hypothesis that inheritance is blending, because we're not seeing yellow-green peas here, kind of intermediate between the two. We're seeing only yellow peas. Now let's just put in a bit of nomenclature here. The first generation that we've got up here is called the parental or the P generation. P stands for parental. And this first generation of offspring is called F1. F stands for filial, meaning offspring or son. And so the P generation when crossed gives you the F1 generation. So the P generation when crossed gives you the F1 generation, and we see only yellow. Green has disappeared. So is green gone forever? Well, when Mendel took 2 of the P plants from the F1 generation, so one here and then another one from another cross of a true breeding yellow and green plant, and then he crossed those together, what he saw were plants that produced yellow peas only, and also plants that produced green peas only. And because he did this many, many times, he could see that they always followed a mathematical ratio of 3 yellow plants to 1 green plant. So in the F2 generation, green has come back and in a very specific mathematical ratio to the yellow plants. So what did Mendel conclude from this? First, this shows that blending is not the right model. First, we don't see yellow-green here, but more importantly, green, which looks like it's disappeared in the F1 generation, comes back in the F2 generation. So it hasn't been blended out. It's been hiding in the F1 generation, concealed somehow. What Mendel developed as his model was that there were heritable factors or units of inheritance, and we now call those genes, but that was a term that came later. And these heritable factors pass unchanged from parent to offspring. And from his observations and careful thinking, he developed the hypothesis that each parent has 2 copies of each of these heritable factors, but only contributes 1 copy to the offspring. And he called this the law of segregation. So a parent might have a big Y and a little y heritable factor, but to the offspring it can only give either the big Y or the little y in its gametes, so these might be its gametes. We now know that this is a result of genes being on chromosomes and the two chromosomes of a homologous pair separating from each other in meiosis as gametes are formed. But Mendel didn't have the benefit of that, so he was deriving this hypothesis in the absence of that just from his experimental data and careful thinking. And Mendel also deduced that the phenotype, the outward characteristic, like yellow or green peas, is determined by the combination of what we would call alleles, although he didn't use that term. So looking at one of his experiments, Mendel deduced that both of the alleles, or both of the forms of the heritable factors, as he might have said, in the pea generation were the same because they were true breeding. They were homozygous. So we'll call this one big Y, and this one little y, and I'm going to show my little y's trailing out the bottom here to distinguish them because y's look kind of similar. Now, each of these can contribute only one kind of gamete. The gamete that this can contribute is a big Y gamete, and the gamete that this one can contribute is a little y gamete, so either a sperm or an egg. And when those fertilize to create the F1 generation, we get big Y, little y. That's our only option. And then when we cross this with another of the same generation, which is also big Y, little y. In this case now, each of these can produce two different forms of gamete, one with the big Y allele, one with the little y allele. And so when we put those together in all of the possible combinations, we see that one of those combinations is little y, and that is what Mendel deduced was happening in the green pea over here, whereas big Y, and big Y, little y account for the other three possible combinations. There's two big Y, little y combinations and one big Y combination. And it's because of that that we get this ratio. We'll look at this more in the next lesson as we look at Punnett squares. And in explaining what was happening here as the green disappeared and came back, Mendel introduced the concepts of dominance and recessiveness, which we can use to describe traits or alleles. And he would say that yellow, or the yellow allele, the big Y allele, is dominant to green, or the little y allele. So that in the heterozygote here, when you have one of each of them, we see the phenotype associated with the big Y allele, namely yellow. And only when we have just little y's do we allow the phenotype that has receded into the background, the green phenotype, to come back out. So dominance and recessiveness help us understand what's going on here. And we determine which allele is dominant and which is recessive by looking at the phenotype of the heterozygote. So in the case above, when we had a big Y and a little y, we saw that we had a yellow pea. And that's the phenotype associated with big Y. So we would say big Y, or yellow, is the dominant allele or the dominant trait. And so that's a first look at Mendel's experiments and how they helped him develop a particulate model for inheritance.